Hello everyone, my name is AJ Keefe. In this lecture I'll be covering adult neurogenesis and fatty acid oxidation. I'll apply these concepts to real-world research by reviewing this recent paper titled "A Fatty Acid Oxidation-Dependent Metabolic Shift Regulates Adult Neural Stem Cell Activity," published in the Journal of Cell Metabolism. I recommend that my viewers pause the video at each new slide and read through the material before playing my commentary. And if you have any questions, please comment below and I'll be sure to respond. Thanks for watching. When talking about adult neurogenesis, we are talking almost exclusively about these two parts of the brain called the subventricular zone and the subgranular zone. And neurogenesis at these two locations results in the integration of new neurons inside the hippocampus. This is the hippocampus right here. And the hippocampus is essentially the site of long-term memory in our brain, and it's also the primary site of new neurons in the adult brain. Now, I'm sure there's probably very important differences and distinctions in how neurogenesis occurs between the subventricular zone and the subgranular zone, but I don't think I have the time to differentiate and explain both of them. So I'm just going to be discussing neurogenesis as if it were both processes were interchangeable. But just remember that there are probably distinct and unique pathways associated with the subventricular zone versus the subgranular zone. But regardless, adult neurogenesis in the hippocampus from either the subventricular zone or the subgranular zone has been shown to be very important for memory and mental health. And the prospect of harnessing or modulating neurogenesis could prove therapeutic for many different disorders. One of the more promising avenues of neurogenesis research in terms of therapy is probably depression, because studies have shown that depression is marked by decreased hippocampal brain volume and decreased neurogenesis. And in general, inhibiting neurogenesis in mice has been shown to cause memory deficits, symptoms that resemble depression and anxiety in mice. And it has also been shown to increase the rate of progression of a mouse model of Alzheimer's disease. So adult neurogenesis is no trivial matter, and understanding how it works can be very beneficial to a wide range of diseases. So the neural stem cell precursors that give rise to neurons are these quiescent, meaning non-proliferative, radial glial-like cells in the subventricular and subgranular zone. And they can be activated by various cues to differentiate into neurons or astrocytes or many different cells. So again, the precursor cells are called radial glial-like cells, sometimes also simply called neuronal stem precursor cells, or NSPCS. And they are quiescent, non-proliferative, until activated by various molecular cues. So the neural stem precursor cells, or the radioglial-like cells of the subgranular zone, are marked by the expression of SOX2, SOX2, GFAP, nestin, and SPOT14. And the maintenance of these quiescent neural stem cells is achieved by BMP, right here, bone morphogenic factor, GABA, and Notch signaling. So SOX2 is probably the most important factor to note in the maintenance of quiescence, because it's a transcription factor that upregulates many of the genes that are critical for the stem cell state. And SOX2 is kind of ubiquitous across many different stem cells, and it's universally kind of recognized as being basically synonymous with the stem cell state. SPOT14, SPOT14, is another critical protein that is important for this discussion because it modulates lipid metabolism, and we're going to discuss that in the next slide. And the maintenance of neural stem cells is again reliant largely on BMP, or bone morphogenic protein, and its downstream signaling pathways, and also the inhibitory neurotransmitter GABA. So the activation and the proliferation of radial glial-like cells is marked by the expression of DCX, the function of which is not necessarily important, but the expression of DCX marks the exit from quiescence and the entrance into proliferation. The transition is induced most critically by WNT, or Wnt signaling, and IGF signaling pathways. And eventually, when these proliferating glial-like cells become neurons, they are marked by the expression of NEUN, N-E-U-N, which is a very common and widespread marker of neurons that you see stained for in many different research papers. What's interesting about stem cell division is that it can actually happen in two different ways. So activated radial glial-like cells can divide symmetrically, giving rise to new radial glial-like cells expressing SOX2 and SPOT14, or radial glial-like cells can divide asymmetrically, giving rise to proliferative neuronal precursor cells expressing DCX. So in other words, not all radial glial cell divisions will lead to neurons. Sometimes they divide simply to replenish the stem cell population. Not only that, but these radial glial-like cells are pluripotent. So sometimes they can give rise to cells like astrocytes or oligodendrocytes or microglia. And furthermore, only very few neuronal precursor cells actually survive to become full-fledged mature neurons. So proliferating neural stem precursor cells are under very stringent surveillance by microglia, which mediate a process called pruning, or selective, basically selective killing and selective apoptosis and selective phagocytosis. So there's actually, in fact, 80% of newborn neurons derived from these radial glial-like cells end up entering apoptosis and are phagocytosed by microglia. So the journey from quiescent radial glial-like cell to neurons is marked by the loss of SOX2 and SPOT14, the gain of DCX and NEUN, and nearly 80% of newborn neurons are culled and pruned and destroyed before they become mature neurons. Another important metabolic concept that we need to address in order to understand this research is fatty acid oxidation and fatty acid synthesis. So I have both these processes, fatty acid oxidation and fatty acid synthesis, roughly divided into these two sections, low energy demand and high energy demand. During low energy demand, acetyl-CoA, right here, acetyl-CoA derived from pyruvate from glycolysis, or citrate, the citric acid cycle, is converted into long-chain fatty acids by fatty acid synthase. Right here, fatty acid synthesis is done by fatty acid synthase. But before this can happen, acetyl-CoA needs to be activated. It needs to be primed for the addition into those fatty acid tails. And this is done by the ACC enzyme right here. ACC activates acetyl-CoA, which turns it into malonyl-CoA. And malonyl-CoA is the actual substrate that gets polymerized onto into the fatty acids. And malonyl-CoA, the precursor to fatty acid synthesis that is derived from acetyl-CoA, also inhibits a transporter called Cpt1. Cpt1 we'll discuss in a second, but it's a fatty acid transporter. So during fatty acid synthesis, we have acetyl-CoA being activated by ACC, which turns it into malonyl-CoA, which both is the substrate for fatty acid synthase and simultaneously inhibits Cpt1. During high energy demand, to get those extra ATP molecules, a cell will take these long chain fatty acids right here, and it will shuttle them into the mitochondria through these transporters Cpt1 and Cpt2, where they enter fatty acid oxidation, or FAO, also called beta oxidation. And this process releases the acetyl-CoA that is bound up in those fatty acid tails. And then the acetyl-CoA, of course, enters the citric acid cycle, which then creates ATP. And it releases the high energy electrons for oxidative phosphorylation. So CPT is the transporter and also the rate limiting step in fatty acid oxidation. So this little transporter right here is actually the rate limiting step in how quickly fatty acids can be degraded. Cpt1 import of fatty acids is the rate limiting factor in fatty acid oxidation. It's very important. The inhibition of Cpt1 by malonyl-CoA, right up here, prevents what's called a futile cycle. A futile cycle is when something is being created and degraded at the same time inside of the cell. In the context of fatty acids, a futile cycle would result if CPT was not inhibited by malonyl-CoA. Because if in this alternative universe, if malonyl-CoA didn't inhibit CPT1, then the fatty acids that get made by fatty acid synthase would simply get imported and then degraded through the citric acid cycle. And then these acetyl-CoAs could then leave the mitochondria, form more malonyl-CoA, make more fatty acids, then get degraded again. So that would be a futile cycle. So by making the precursor to fatty acid synthesis, malonyl-CoA, simultaneously inhibit the rate limiting step in fatty acid degradation, CPT, the cell avoids a futile cycle. So just to summarize, fatty acid oxidation goes like this. Cpt1 mediates the import of long-chain fatty acids into the mitochondria, where they are completely oxidized into acetyl-CoA, or partially oxidized into acetyl-CoA, then completely oxidized in the citric acid cycle. In contrast, during fatty acid synthesis, acetyl-CoA is activated and converted into malonyl-CoA by the enzyme ACC, which inhibits CPT1 to prevent a futile cycle. Malonyl-CoA is thus the building block for fatty acid chain elongation and the inhibitor of fatty acid oxidation. Okay, so now that we've reviewed neurogenesis and fatty acid metabolism, now let's put these two concepts together. So fatty acid oxidation is uniquely important for neuronal stem cells. In fact, the metabolism of those radial glial-like cells of the subgranular zone is pretty much dominated by fatty acid oxidation, and it's believed that the expression of this protein, SPOT14, is what mediates this choice of metabolism. The SPOT14 protein is upregulated in the quiescent neuronal stem cells, those radial glial-like cells, and it appears to promote fatty acid oxidation by inhibiting ACC. So ACC is the enzyme that activates acetyl-CoA, and it converts it into malonyl-CoA. So SPOT14 mediated inhibition of ACC prevents acetyl-CoA from being activated and prevents malonyl-CoA production. So just to reinforce that idea, in neuronal stem cells, this SPOT14 protein inhibits malonyl-CoA production by inhibiting the enzyme ACC, which normally converts acetyl-CoA into malonyl-CoA. And since malonyl-CoA is the building block of fatty acids, and it's also what inhibits fatty acid oxidation. Blocking its production promotes fatty acid oxidation, and it inhibits fatty acid synthesis. So in quiescent stem cells, fatty acids are not built, but rather they're imported into the mitochondria through this CPT transporter, and then it gets completely oxidized through beta oxidation and the citric acid cycle. However, following activation and during proliferation of these stem cells, the neural stem cells increasingly begin to rely on glucose and glycolysis for ATP, which makes sense because neurons, if you remember, are almost completely reliant on glycolysis and glucose for their ATP, and they don't actually have much of an interest in lipids. So the downregulation of SPOT14 is speculated to be at least partially responsible for this change in metabolism by allowing ACC to begin producing malonyl-CoA from acetyl-CoA. So the sudden activation of ACC, which is actually done through this intermediate called MIG-12, but either way, the activation of ACC, because SPOT14 is absent, leads to the production of malonyl-CoA, which both inhibits CPT1, so inhibits fatty acid oxidation, and also activates fatty acid synthesis because it's the precursor to longer chain fatty acids like palmitate. So again, the loss of SPOT14 allows ACC to activate acetyl-CoA and convert it to malonyl-CoA. And then malonyl-CoA then leads to fatty acid synthesis, and this occurs as a stem cell is transitioning into a neuron. Okay, so given all these ideas, let's finally get into the actual paper. The impetus for this research was based on previous work that was done by this same lab that found that proliferating NSPCs, or neural stem precursor cells, were synthesizing and accumulating lipids, and this process was actually critical for their survival and differentiation into neurons. However, the role of fatty acid oxidation, the counterpart of fatty acid synthesis, is poorly understood. So the current paper asked the question, does the oxidation of fatty acids play any role in adult neurogenesis? And this is in the context of a brain that is almost exclusively glycolytic. So again, remember that brain is almost exclusively glycolytic and reliant on glucose. So there really is not much research on the role of fatty acids in the brain. So here are the results of the paper. So first they observed that quiescent neural stem precursor cells in culture had high levels of fatty acid oxidation. And the rate-limiting factor of fatty acid oxidation, CPT1A, which is the subunit of CPT, is highly upregulated. To confirm these findings in vivo, in the live animal, they did some immunohistochemistry, and they found that CPT1A is indeed expressed in the radial glial-like cells of the subgranular zone, which is quite fascinating because the only other cells in the brain that express this critical lipid transporter are astrocytes. Next, they used a very fancy tamoxifen inducible Cre-Lox system to delete CPT1A in only the NSPCs, and they observed that this greatly disrupts neurogenesis by not only reducing the number of clones, but also changing the outcome of those divisions. So instead of producing neurons, many of the NSPCs were dividing symmetrically to regenerate more stem cells. And lastly, they supplied NSPCs with malonyl-CoA exogenously. So in culture, they just gave them a bunch of malonyl-CoA, and this precursor to fatty acid synthesis in the CPT1 inhibitor was alone able to induce proliferation. So this malonyl-CoA precursor alone was able to induce proliferation. To determine if fatty acid oxidation is important for quiescent NSPCs, they looked at a couple different markers. First and foremost, the levels of CPT were assessed, which again is the rate-limiting factor in fatty acid oxidation by regulating the import into mitochondria. And in this Western blot, we see that the amount of CPT1 is vastly reduced during proliferation, and it's very high during quiescence, indicating high fatty acid oxidation during quiescence and a subsequent downregulation during proliferation. In the middle, we see the results of an experiment where they supplemented the cells with radioactive palmitate, which is a lipid that is readily taken up by cells, and they observed that the resulting CO2 that was released was radioactive in quiescent cells, but there was much less radioactivity in proliferating and previously quiescent cells. So in other words, quiescent cells are taking up the radio-labeled palmitate and then spitting out radioactive CO2, indicating it was metabolized, while proliferating and ex-quiescent cells are doing much less oxidation of palmitate. And then finally, they're looking at the actual building block of fatty acids. We see, which is malonyl-CoA, of course. We see that proliferating cells have high levels of malonyl-CoA. So when they're proliferating, they're synthesizing lipids, and when they're quiescent, they're eating lipids. And also, I don't have the data shown here, but when they inhibited fatty acid oxidation in quiescent cells, it basically was causing cell death. So the import in the fatty acid oxidation in quiescent neural stem cells is a very critical process. Next, the researchers confirmed that the upregulation of CPT1 was occurring in vivo, and so they generated a transgenic mouse with GFP green fluorescent protein under the CPT1A promoter. So wherever you see CPT1A expression, you will also have GFP expression. We see robust GFP expression throughout the subventricular zone, the subgranular zone. So this is the hippocampus right here, and this is the subventricular and the subgranular zones. But regardless, what they're saying is that CPT1A expression is very high, and it's largely co-localizing with SOX2 and nestin. So SOX2 and nestin in red are the markers of the quiescent stem cells, and it's largely co-expressed with CPT1. And they did some more immunohistochemistry that was showing that CPT1A expression is absent in DCX proliferating cells. So remember, DCX is expressed in proliferating and differentiating NSPCs. So just to summarize, they found that CPT1A is expressed in quiescent SOX2 positive cells. So we see the amount of GFP positive cells that are also SOX2 is almost 100%. So wherever you have SOX2 expression, you also have GFP, indicating CPT1A expression. In contrast, wherever you have DCX expression, you have no GFP expression. So they're inversely related. If you have DCX expression, you don't have GFP expression. Next, the researchers deleted CPT1A in neuronal stem cells using an inducible Cre-Lox recombinase. Since the recombination efficiency of this particular setup was rather low, they were able to identify specific cells in their lineages by the gain of yellow fluorescence, which through fancy genetic engineering is only expressed in cells with active Cre. So I won't get into the specifics, but what they found is that in Cpt1a deleted NSPCs, they were much less proliferative, where only about 50% were producing more than one clone, compared to 75% of wild-type NSPCs producing more than one clone. So in the clone size distribution, we see in the wild-type cells that only about maybe 25% are only producing one, but in the Cpt1a deleted cells, 50% are only producing one. We see this more proliferation in the wild-type cells. Furthermore, the majority of Cpt1a deleted stem cells were dividing symmetrically. About 80% were either producing only radioglial cells, indicated by R. So down here, 80% were producing either radioglial-like cells or no progeny at all, indicated by the X right here. So sorry, I'm looking at this bar right here. So 80% are either producing only radioglial-like cells or neither, or any cell. And only about 10% of the Cpt1a were producing neurons. So we see right here, only about 10% are producing neurons compared to 35% in controls. So we see the clone composition is disturbed. There are less neurons being produced and more symmetrically divided radioglial-like cells. And we also see overall there's less division in the Cpt1a deleted cells. Since malonyl-CoA plays such a major role in not only building fatty acids, but also stops fatty acid breakdown by inhibiting Cpt1a, they wondered if malonyl-CoA alone was capable of inducing proliferation. And indeed, they found that exogenous supplementation of malonyl-CoA was capable of inducing quiescent stem cells into proliferation, as measured by Ki67 positive cells and phosphorylated H3 histones. So these are just two very well-known markers of proliferation. And you see a dose-dependent increase in the amount of proliferation when malonyl-CoA is added, which is really quite amazing because malonyl-CoA is just a metabolite of acetyl-CoA. It's really not a very drastic thing to add to a cell culture, but it produced a pretty substantial increase in proliferation. So in other words, the inhibition of Cpt1a, the inhibition of fatty acid oxidation in a physiologically relevant context, actually induces quiescent cells into proliferation. So it might actually be the Q that induces proliferation of stem cells. So our summary is that quiescent neural stem cells express high levels of Cpt1a and show all the hallmarks of increased fatty acid oxidation. Deletion of Cpt1a in neural stem cells reduces their proliferation and it disturbs their proper differentiation into neurons. The upregulation of malonyl-CoA may underlie this metabolic shift that accompanies the proliferation as it inhibits Cpt1a and fatty acid oxidation. So they didn't actually look at spot 14, but speculated that spot 14 normally keeps Cpt1 active, it keeps fatty acid oxidation active because it prevents malonyl-CoA from accumulating. However, when spot 14 is lost, ACC begins making malonyl-CoA from acetyl-CoA, and when malonyl-CoA accumulates, because ACC is now active, because spot 14 is gone, it then inhibits Cpt1a, it inhibits fatty acid oxidation, and lipogenesis and fatty acid synthesis may be the cue that induces proliferation of neural stem cells. So I hope you guys enjoyed the video, and if you have any questions or need clarification, please comment, and thanks for watching.